there has been a fair amount of in vitro work done regarding Nedd4-1 and Parkinson's disease, particularly. So Nedd4-1 in the past has been shown to ubiquitinate alpha-synuclein in cell culture, and it's also been shown to be upregulated in the substantia nigra of Parkinson's disease patients. So what we were interested in is what's been shown in vitro also the case in vivo, and also what might be the role of Nedd4 ubiquitin ligases just generally in the midbrain, because that's not really been looked into too much. A lot of Nedd4 work has been done in the cancer field, and some of it's been done in the epilepsy field, but actually nothing really in neurodegenerative diseases to speak of, particularly in vivo. So that's why we use our Nedd4, Nedd4-1, Nedd4-2 dopaminergic-specific knockout mouse model to actually examine what might be the case. And that's an ongoing analysis that we've had going on throughout my PhD, really. So that was, I guess, really the background. Nedd4-2 is not so well researched, so we also wanted to look into that, which is it's a related ligase to Nedd4-1. So we did it actually in a couple of mouse models, which we generated in-house. So we've got one which is under the dopamine transporter and one which is under the tyrosine hydroxylase promoters. So we've got two different ways of deleting our ligases. What I found actually was that the DAC-Cre system, which is the dopamine transporter-driven Cre system, to delete those two ligases was actually the most efficient way to delete them. So then I then kind of looked at what effect that might have on the midbrain system itself. So particularly with regards to development and maintenance. And what we found actually is that Nedd4-2 seems to be quite important there. Nedd4-1, not so much so. Then we took a second approach using that TH-driven Cre to delete both Nedd4-1 and Nedd4-2 together, and actually to look what happens when alpha-synuclein is brought into the situation as well. So we used an A53T mutant alpha-synuclein, which is expressed in the same cells. And that actually meant that we saw the interaction then between Nedd4 ligases and alpha-synuclein together in vivo. What we found was that in aged mice, Nedd4-2 seemed to be more important for the maintenance of the dopaminergic system. So that means that in theory, Nedd4-1 could be important too, and Nedd4-2 seems to be the most important one. So now what I'm doing is trying to find out why that is. So we don't know why that is, and I'm looking at a few different candidates to try and figure out why Nedd4-2 is important and actually what the mechanisms at play upstream and downstream signaling wise. With regards to alpha-synuclein, I'm now going to see if actually these proteins are interacting in vivo. So are they binding each other? Is there more ubiquitination? And is that why we're seeing the results that we did? And also, what are the levels of alpha-synuclein, and why is that happening in these mice? So like, why are Nedd4-1 and Nedd4-2 coming into play with alpha-synuclein in Parkinson's disease?